Good morning, everyone. Thank you for joining. We're just going to wait about 2 more minutes just for everyone to settle. Thank you. Okay. So good morning, everyone. Thanks for joining us today. We are pleased to welcome you to our Realizing Healthier Pregnancies Research Cluster. Thanks for joining us. My name is Erin Showbloom, and I'm a PhD student in epidemiology, as well as a grad research assistant under WICRI member Dr. Stephane Montesanti at the School of Public Health at the University of Alberta. I'm pleased to serve as the session moderator for our presentations today. And on behalf of WICRI, thank you for taking time to attend this session. Just going to share my screen here. Okay, so I want to begin in a good way by acknowledging that the conversation we're having today is taking place on Treaty 6 territory, the traditional lands of the Nehiyac, Dene, Nakota Sioux, Saulteaux, and Blackfoot nations, as well as the homeland of the Métis Nation. We honor the profound wisdom, knowledge, and experiences of the Indigenous peoples who have been stewards of this land since time immemorial, and whose teachings continue to illuminate the path forward for our sacred responsibilities as health researchers now and for generations to come. Okay, I'd also like to thank WICRI's funders, the Stollery Children's Hospital Foundation and the Alberta Women's Health Foundation for making this research cluster possible. WICRI is actively seeking to build this research cluster. Please reach out to WICRI to join the cluster, nominate your collaborators or networks. WICRI's email is in the chat. Michelle from WICRI will stay in this session to answer any questions you might have about participating in the cluster. The session will be recorded and posted to the WICRI website. WICRI will send out the link to the recording afterwards. Please allow 24 hours to access the session recording. So we are looking forward to sharing our research with you and encourage questions after each presentation. And on to the main event. I'm pleased to introduce Dr. Anita Kozyrskyj and Dr. Stephane Bourque. Anita Kozyrskyj is a professor of pediatrics with the Faculty of Medicine and Dentistry at the University of Alberta. She leads the Symbiota Synergy and Microbiota research program on early life shaping of the infant gut microbiome and child immune-related and neurodevelopmental outcomes in the child cohort study. Symbiota was funded by the 2010 CIHR Microbiome Initiative and is now part of CIHR's Impact Microbiome Research Network, where Dr. Kozyrskyj is a co-PI of the Human Cohort Design and Analyses platform. Dr. Kozyrskyj's SIM program has generated 50 papers, 3 book chapters, and trained many next-generation microbiome researchers. Her microbiome papers have received awards for being the most influential and have been cited by position statements on food allergy and neonatal early onset bacterial sepsis. Dr. Stephane Bourque is an associate professor of Anesthesiology and Pain Medicine in the Faculty of Medicine and Dentistry at the University of Alberta, and a Canada Research Chair in Maternal and Perinatal Physiology. Dr. Bourque's research program focuses on elucidating the mechanisms by which developmental stressors such as iron deficiency and neonatal sepsis alter early growth trajectories of the offspring and, in turn, increase susceptibility to cardiovascular and metabolic diseases later in life. He also studies the maternal and fetal effects of novel therapeutics, for example, bioactive compounds, for iron deficiency in pregnancy. His work is funded by the Canadian Institutes of Health Research, Heart and Stroke Foundation of Canada, Stollery Children's Hospital Foundation, and Alberta Women's Health Foundation through the Women and Children's Health Research Institute. So Dr. Kozyrskyj, would you kindly take the floor? Thank you.
Erin, and thanks for that introduction. I'm checking, everyone can see?
Looks great, Anita.
Okay, I'm advancing, that works too, perfect. Wonderful. It's always a pleasure to talk about my favorite topic, my microbiome research, and today I'll be discussing and showing some evidence for critical periods in the establishment of infant gut microbiota in relationship to neural development. First, my Symbiota program derives its samples and data from the child cohort study. And Edmonton is one of the recruitment sites. As mentioned, it's CRHR funded and a big thanks to my current research team and my previous research teams, as shown in the slide, for their contributions in generating the findings that I will show. So my presentation has two themes. One is in relationship to early life, pregnancy, and gut microbiota in the infant, and then how this would affect neural development. Let's just start with an overview of critical periods for the development of our brain. This is a nice review paper, nice visual that shows that the brain architecture in terms of the development of neurons, that critical period is during pregnancy. And during that time, there are some important cells called the microglia, and they are the brain's immune cells that are integral to these processes of laying down neurons and connections. During this time, during pregnancy, we see that the pregnancy gut microbiota changes. So we have a composition in first trimester that is very similar to the non-pregnant state. And there are some changes in the third trimester. You can think of it as changes that prepare for the transfer of maternal microbiota to the newborn. And so some of the changes are related to, for example, increased amounts of proteobacteria, of bifidobacteria being present in that third trimester. In terms of the microglia, we now have a proof of concept that it's interesting that the microglia that we have in our brain, they originate in mid-gestation. So they kind of migrate to our brain during mid-gestation. And this is very much influenced by the presence of pregnancy gut microbiota. So in an animal model experiment, that maternal gut microbiota during pregnancy is required for this to occur. But looking again at this diagram of development, we see that a lot is still going on after birth in terms of the development of the brain, laying down neurons, synapses, and then much later on, the whole process of pruning, where unused or unnecessary pathways are eliminated. And during this time period after birth, we also see a development, and that is the development of gut microbiota. In terms of brain development postnatally, and this is some data from Oliphant and al. In terms of head circumference, which is a good marker for brain development. We see that at around 30 weeks post-conceptual age or post-menstrual age, we see that emergence of trajectories, and whereby you have some infants that have like appropriate head circumference, and then others that the head circumference have started to trail off. And so we see these trajectories emerging like postnatally. And what was noted, found in this paper, is that there are certain factors that determine these growth trajectories of head circumference. And I just want to draw two to your attention. And that is that the way to read this diagram is that you have the factor listed on the left-hand side, and the horizontal bar, and then the length of the horizontal bar indicates the importance, the influence of that particular factor on the trajectory. We see number one at the top that delivery mode is very in these head circumference trajectories, and just below that, also of strong magnitude, is the group of microbiota called the Bacteroidetes. And way down at the bottom, and maybe this is surprising, we see that definitely human milk is influential, but at this point is not as influential as delivery mode or the amount of Bacteroides present in gut microbiota. So what are the normal changes of a gut microbiota in infancy? And we see this pattern. This is from the child cohort study. It's a pattern common to infants worldwide, with some small changes. But basically, we are born in the early weeks and months. Our gut microbiota has a dominance, or relative to later in infancy, that there are higher amounts of the enterobacteria, and they diminish over time. You can think of the microbiota that are present soon after birth as the ones that are more tolerant to oxygen. As the gut becomes more anaerobic, then it creates an environment where the anaerobes thrive. So the Bacteroides are anaerobes, and we can see that their abundance in blue increases in later infancy at the time that the Enterobacteriaceae diminish in abundance. We see a peak of bifidobacteria in relationship to breastfeeding. And then also alongside the Bacteroideaceae, we see the increased abundance of a bunch of Firmicutes, like the Ruminococcaceae, the Violinaceae, the Clostridia. So we see those, and those are also anaerobes. We see them increase over time. And this is like the normal expected pattern. Well, this is the normal pattern if you were born vaginally. As I showed that, head circumference is related to the amount of Bacteroideaceae or Bacteroides, we see then the importance of birth mode and the importance of vaginal birth. Here, I'm presenting in a figure a stack bar plot in relationship to the method of delivery, and this is on the right-hand side of the slide. The relative abundance of microbiota, and then if you just want to direct your attention to the scenarios of cesarean section birth, in orange, we see that the abundance of Bacteroides is really diminished. And this is a pattern that is characteristic in many cohorts, and it's a worldwide pattern. What happens when you're born by cesarean relative to being delivered vaginally. Of course, on the left-hand side, we see that we see the influence of the way you were fed. We see the strong influence of breastfeeding versus not breastfeeding or partial breastfeeding. But the influence of birth mode is independent of a feeding mode in the early infancy period. Like, for example, up until 3 to 4 months of age, and thereafter, what you're fed, how you're fed becomes more dominant relative to the influence of birth mode. Many people refer to this period of the depletion of Bacteroides as the baton has dropped in terms of your trajectory of gut microbiota development in later infancy. Also, some work that my PhD student Sabrun is doing suggests that it's maybe not only birth mode, but perhaps the staying in hospital longer after cesarean section also contributes to this further depletion of Bacteroides. Okay, so let's now transition to look at the evidence of the relationship between infant gut microbes and brain development. Again, both systems are developing postnatally. And first, I want to just talk about some basic main types of composition. And some people refer to these as enterotypes. And based on data from the child cohort study, specifically in relationship to the Edmonton site of the child cohort study. And these results were published in Gut Microbes in 2021. And I will just go over the study population. Now, why we're talking about the Edmonton-based child site is because it is at this site that infants were assessed at 1 to 2 years of age with the Bayley scale. So the Bayley scale assesses cognitive, language, and motor development. And we see that the scores were, on the high side, and that typically female infants scored higher than male infants. This was a population, this is typical of the child cohort study, predominantly high-income families. A quarter were delivered by cesarean section. 16% were exclusively breastfed, a little lower than some of the other child sites. And in these 400 or so mothers and infants, and they were full term, we profiled their microbiota typically with 16S sequencing of V4. We had two time periods, 3 months and 12 months. And then the measures reported there are characteristic of a microbiota study in terms of assessing microbiota diversity, and then looking at the taxon abundance of this is of specific microbes. And then there are some techniques that were used to cluster these microbes to characterize typical compositions. And then finally, there was a method whereby the 16S data was related to a database to predict what likely metabolic functions dominated in terms of this microbiota profile. So that in a nutshell is the study population. And let's go on to look at some of the findings, and I'm going to focus on the findings of gut microbiota clusters, and this is at 1 year. Remember, there were 2 time points, but I'm focusing on 1 year. And we see that there are 3 main clusters. In minority of infants, a cluster that is dominated by the proteobacteria. Then we see that half of the infants, the cluster is dominated by Firmicutes, and then a third of infants, their cluster is dominated by the Bacteroidetes. The Bacteroides cluster, they are more likely to be delivered vaginally, likely to be delivered by cesarean. And again, this is at 1 year. Now, in terms of infant neural development, I want to say that these findings have now been part of a systematic review that was recently published. And I'm just circling our findings there to show the coefficients in relationship to the Bacteroides cluster. This is other microbiota changes reported by other studies. So here, these are violin plots just to show, and I'm reporting cognitive scores of the Bailey scale according to the 3 clusters I just described to you. And so no relationship between the 3-month clusters, which I didn't present to you, and cognitive scores at 1 year or 2 years. There was no association between the 1-year clusters that I did show you and 1-year scores, but we did see an association between the 1-year cluster and 2-year cognitive scores. And this was in terms of the higher cognitive score being seen in the cluster dominated by the Bacteroides. In more detail, we see on this slide, with respect to the 3 domains of Bailey's neural development assessment, cognition, language, and motor domain. And again, these are, I'm reporting to you the statistically significant results at 2 years. In relationship to the cluster, we see that the reference cluster is cluster 1, which is dominated by the proteobacteria. And I'm just highlighting the statistical significant findings in terms of cluster 3, which was dominated by the Bacteroides. And we see that within the all domains that there is like 3 to 5 point higher score for cognition if the infant had this dominant cluster being of the Bacteroides type. And we see that the results are similar with crude and adjusted for those variables reported on the screen. Now, knowing that there was a difference in scores, cognitive scores between male and female infants, we conducted a stratified analysis. And then we see that, first of all, the results were no longer statistically significant for female infants. And so all of the statistical significance was in the male infant. And that we see that for both cognition and language, that having the Bacteroides clusters increased those scores 6 points for cognition and 8 points for language. Now, I'm bringing you back to some basic epidemiology in terms of the Bradford-Hill criteria for disease causation. And I'm going to argue how in this particular study fulfilled some of those criteria, starting with temporality. So I did note that the microbiota profile was 1 year, and the association was with cognition at 2 years. So that does indicate temporality. However, just with, one time point, we don't know if that cognitive score was already present at the time the microbiota sampling or the microbiota were profiled. So much better to have 2 time points, a pre and post. And in fact, we did. So we were able to determine the difference in scores between 1 and 2 years in relationship to the microbiota clusters. And we see here again for male infants that indeed statistical associations were observed for the cluster 3, increasing the cognitive scores by almost 5, and then the language scores by 6. So this is a little bit more stronger evidence in terms of temporality relating the microbiota cluster type to the change in cognitive score. Now, what about some of the other requirements of Bradford-Hill? For example, dose response, which is one of, if there is evidence of a dose response, it's definitely is stronger evidence for a causation pathway. And indeed, for some of the variables, for example, the relative abundance variable of a particular microbe at the species level, we found that increasing amounts or abundance of the Bacteroides, and we have the Fragilis is the particular genus that with increasing amounts of Bacteroides fragilis, we did see an increase score under the cognitive domain. We also saw increased scores for the Bacteroides uniformis for cognition and language. So again, another a criterion support of a possible causation pathway between the change in microbiota and the neurodevelopmental score. I mentioned that one of the analyses involved a comparison of the 16S sequences to a database that predicts metabolic function. In this particular figure, I'm showing you the metabolic functions that were higher with this Bacteroides cluster. I want to just focus in on the ones related to sphingolipid biosynthesis. So they were more dominant with the Bacteroides cluster in comparison to the other clusters. So then let's some of the other Bradford-Hill criteria in terms of the results of this study. So I'm happy to report that there is some consistency with what has been reported in the literature to date, in particular the Carlson and all study which we reproduced. This is no small feat because unfortunately, the microbiota literature is filled with inconsistent findings. Maybe it's not surprising, it is a complex system. So when you actually have a finding that is consistent with findings from another cohort study, that is an indication that there is a strong signal and maybe a pathway there. In terms of plausibility, now I want to talk a little bit about biology and microbes. So not all microbes are alike. The Bacteroides are ones that have a more sophisticated metabolic capacity. They actually can make these sphingolipids from substrates in breast milk, or they from their own short chain fatty acids produce, for example, propionate. So they can produce these sphingolipids, which then are absorbed and are utilized by the host. So the host doesn't care whether the source of the sphingolipid is from the human cell or produced by microbiota. And these sphingolipids are really important in terms of neurogenesis. They're important in terms of myelination of the axon. Now, another interesting point is that these substrates, like for example, sphingomyelin, they're provided by breast milk, and then we know that there are associations between breastfeeding and higher neurodevelopmental scores. There's also some evidence that with increased amounts of this sphingomyelin, you do see improved myelination of the brain. So there's some biological plausibility in terms of our findings related to the Bacteroides and the capacity to manufacture sphingolipids. So so those are the main findings from this Tamana paper based on the Edmonton site of the child study. I just want to go back to the findings that were not statistically significant. I mentioned that there were no associations between the early life gut microbiota profile at 3 months and then 1 or 2 year scores. So we have kind of revisited those results, and so whereas there were no differences in of the cluster types, we also have data on metabolites measured in fecal samples. So specifically, if they're short chain fatty acids, we know they're produced by microbiota. We have more detailed information on a very interesting microbe, C. difficile in infancy, because it's not present in all infants. When it's present, it doesn't produce the same GI symptoms as it would in adults. And breastfeeding reduces the amount of C. difficile. So at 3 months, and this is a paper that we are submitting, we actually do see some relationships between a short chain fatty acid propropionate and then C. difficile abundance in terms of breastfeeding and cognition. So exclusive breastfeeding reduces C. difficile, it reduces propionate, and this improves cognitive scores. And with that, I will be finishing with just this overview diagram of what I think is happening in terms of the Bacteroides, starting with its composition in human adults, and that we see that there is a worldwide variation. Of course, then there would be could be some changes during pregnancy in terms of diet or stress, and then with the next stage in terms of method of delivery, whether it's cesarean or vaginal, we see an opportunity for the Bacteroides to be depleted. And then that sets up a trajectory where we have lower Bacteroides initially, and they remain that way in later infancy, which then, according to our data, would lower some of the neurodevelopmental scores. Breastfeeding has a role in terms of its provision of some of the substrates. I mentioned sphingomyelin, which could be used by the Bacteroides in terms of the production of sphingolipids, which are important for neurogenesis. So we're still at very early stages in terms of understanding these complexities between gut microbiota development in infancy and neurodevelopment. But with the study I have shown you, now it's, in conjunction with others that have been published, we're starting to see the evidence. And with that, I like to finish with another slide of my team, and the results that I showed you that are, we're now looking at the 3-month gut microbiota. That was the thesis of Carmen Tessier, who's standing next to me in this photo, which was, I believe, taken at Wickery Day. So thank you, everyone, and I welcome your questions.
Thank you very much, Dr. Kozyrskyj. As a student of epidemiology, I really appreciated how you connected your research to the Bradford Hill criteria. So now I'm going to invite everyone to ask your questions. If you could, please use the raise your hand feature to ask a question, and I'll do my best to invite you to voice your question in the same order that hands are raised. If you're more comfortable using the chat feature to ask your question, we'll do our best to voice your question for you. If we get the substance of your question wrong, please unmute yourself and help us to get it right. And yeah, we're looking forward to your questions, so please don't be shy.
I do have a question.
Yes, please, Maria, go ahead.
Thank you, Anita, for a wonderful presentation. I was very intrigued about the findings about breastfeeding and the infant microbiome, and there's a lot of interest in that relationship. And I was wondering if you can help us to understand what are some of the key controversies or debates around that question, and what areas from your perspective might need further exploration between the relationship of breastfeeding and infant microbiome and neurodevelopment.
As reported from the child study, some of the nuances of breastfeeding, whether feeding at the breast or, or feeding pump milk, that has been explored. And so that does cause some differences in composition. Like, for example, now, in terms of neural development, I think that that literature or that those studies are forthcoming. I think it's important to note, and I pointed this out early on, that in early infancy, the breastfeeding influence is independent of the birth mode influence. So you'd see the same pattern of Bacteroides depletion, even in the exclusively breastfed infant. And the influence of breastfeeding is stronger in later infancy.
Okay, thank you.
Thank you for your question, Maria.
I'm happy to answer basic questions about infant gut microbiota.
Hello, can you hear me?
Yes.
Yes, we can.
Dave Olson. Wonderful talk, Anita. It's probably the best microbiome talk I've heard.
Oh, well, thank you.
I'm curious, though. A lot of the evidence is correlational. Is it possible to do cause and effect experiments? Can you add sphingolipids to babies and make them better babies?
Well, you're talking about maybe some experimental evidence.
Yes.
Well, I think before going there, I wanted to point out, and maybe I didn't make this clear, that I showed towards the end, I showed some of these triangles.
Yep.
So what's becoming increasingly popular, and we had initiated this in the Symbiotic program early on, is to conduct mediation analysis. So if we're interested in the relationship between X and Y, and you want to know whether M, like gut microbiota, composition or any other indicator is involved in the pathway, it could be like in a causal pathway, you can pursue mediation analysis. Now, I think this would be the next step, because you have as your starting point these association studies, and then the next is is really any characteristic of gut microbiota, like the metabolite or particular microbe, is it involved in the pathway? I recommend that as the next step. And of course, then after that, you have the experimental evidence, which today much of it's done in mouse models and then maybe some piglet models. The issue with that is that mice gut microbiota are not identical to the human gut microbiota. So you can, these studies can be proof of principle to show if you change that, you and see also a change in your outcome. So the change was brought about through the change of gut microbiota. Their species, they're not, the best model to pursue. Now, what you're suggesting is more of like an interventional study, right? Our randomized controlled trial in an infant, mom, etc. Those are, I think, are a bit challenging to do, right? Because one wants to be careful what they're deciding to feed infants, in terms of testing their effectiveness. Oh, I think it's a bit challenging. I know there's interest in testing different probiotics. Some of the results have been disappointing. I think it's because we don't have a proper understanding of the role of different kinds of microbes, like in the time period after birth. That if we feed an infant a lactobacillus probiotic, that we would expect to see a change where, maybe at that time point, they're not one of the, they're not the only critical, microbe.
Thank you very much.
Thank you, David, for your question. Are there any further questions? If not, we'll have some time after Dr. Bourque's presentation to ask questions again.
Perfect, and I'll stop sharing.
Okay, thank you again, Dr. Kozyrskyj. So, Dr. Bourque, if you could do us the honor.
Can you hear me okay? Yeah. Perfect.
Yes.
Well, first of all, Erin, I want to thank you for the kind introduction at the beginning, and I especially want to extend my thanks to Michelle and the rest of the WICRI team for the invitation to speak here today on a topic that's near and dear to my heart, which of course is iron deficiency anemia in pregnancy. And I thought, given the audience in the venue, I would talk less about some of our preclinical studies and more about some of the public health challenges, and talk about why or I guess my group in general, tend to think about this as a deceptively complex global health issue. Okay. So anemia is a condition that's characterized by either a reduction in red blood cells or a reduction in the concentration of hemoglobin within those red blood cells. And it's, by extension, it's a condition that's associated with a reduced oxygen carrying capacity within the blood. It affects anywhere between it affects anywhere between 20 to 25% of the global population. And what's particularly interesting in the context of our work, but I guess also in a global health perspective, is that pregnant women are most at risk. Pregnant women and young children and infants. And I'll tell you why that is in just a second. But in general, the World Health Organization estimates that about 30%, 38% of all pregnant women are anemia or will develop anemia at some point during their pregnancy. And in fact, more recent studies suggest that this might actually be an underestimate. And we know that iron deficiency and anemia are associated with a number of adverse pregnancy outcomes. So it increases the risk of perinatal death, congenital defects, preterm birth, and even in the absence of such severe complications, it's also associated with fetal growth restriction, which again, as we know in the go-ahead context, has important implications for long-term health of the offspring. So this is a map by the World Health Organization showing the prevalence of anemia specifically in pregnancy. And so you can see that most countries fall somewhere between 20 to 40%. There's a few notable exceptions, including countries in Southeastern Asia, as well as countries in sub-Saharan Africa. And in these countries, prevalent rates can go as high as 80% in some cases. Okay. The other notable exception is the US, and you might be convinced here that the US is doing a lot better, but it turns out actually that the prevalence there is somewhere between 18 to 19%. And so it's not that far removed from Canada's prevalence of about 23 to 25%. So iron deficiency and anemia are often conflated, and it's important to remember that in anemia, there's a number of causes, and most of them are listed here. You can imagine that there's quite a lot of geographic heterogeneity as well as demographic heterogeneity. And so this is a really nice study that's published that shows the causes of anemia based by age group. And so you can see that the vast majority of very young children, the anemia causes are attributed to iron deficiency. Okay? And the other subgroup that we're particularly interested in, again, as I mentioned, is pregnant women. And you can see again that the vast majority of cases is also attributed to iron deficiency. And I think it's really important because obviously understanding the underlying cause is critical to managing and managing treatments. And so if, for example, the anemia is caused by thalassemias, where there's actually, which is actually associated with iron loading, well, in those circumstances, administration of iron supplements can actually be deleterious to health. Okay, so really understanding the underlying causes is critically important. But why is iron deficiency so common in pregnant women? So this is a graph that shows the nonlinear time course. And on the y-axis, you see the iron demands. And so under normal circumstances, iron loss accounts for about 1 milligram per day. However, in women of reproductive ages, the iron demands go up quite a bit because of the periodic blood loss. Okay, but during pregnancy specifically, and specifically during the second and the third trimesters, we see a massive increase in iron demands. And a lot of that is driven by increasing blood volume expansion in the mother, as well as increasing demands from the fetus and the placenta. And so the second and third trimester, iron demands can go up almost 7-fold compared to normal. And I think it's worth mentioning here that because of menstruation and sort of poor absorption of iron in general, many women of reproductive age enter pregnancy with depleted iron stores to begin with. So that you can imagine that, you know, as the iron demands increase as pregnancy progresses, it's really hard. Okay. So the other aspect that's important to distinguish between iron deficiency and other causes is that whereas anemia is generally associated, as I mentioned at the beginning, with a reduction in oxygen transport, iron deficiency superimposed on that is associated with a number of other biochemical alterations. And so every cell in the body uses iron in some capacity, and these are some of the major processes for which iron is involved. And so then you can imagine then the combined effects of iron deficiency, which impacts these biochemical functions with the impaired oxygen transport, makes for an entirely separate entity from just anemia due to other causes. Okay, so it's important to sort of keep that in mind. But nevertheless, you know, having said that, I still think it's a bit surprising that iron deficiency is so prevalent. And that's one of the reasons why in my lab, we tend to think about this as a iron deficiency and anemia as deceptively complex global health issues. And the reason why we say that is because, you know, at first glance, it seems like a relatively simple and straightforward problem. It's reasonably easy to identify with simple blood tests for anemia. And the other aspect is that we know what the problem is. It's a lack of iron, and there are iron supplements available. So the solution seems straightforward. Okay, but it's not altogether clear why it is so prevalent and in fact so intractable. Okay, and it turns out actually there, in terms of public health challenges, there's quite a lot of challenges or quite a lot of difficulties. And I'll just go through the diagnosis and the treatment pieces separately. And then I want to get into some other preclinical data that we've collected. So the first one is the diagnosis piece. And, so in the progression of iron loss, which typically occurs over chronic periods of time. So this usually manifests over the course of months to years, okay? And so in the initial stages, what happens is that we don't see an impairment of hemoglobin levels. The first thing to go down is iron stores. And you see this gradual depletion of iron stores, as well as the concomitant reduction of ferritin, which is the primary iron binding or the iron storage protein, okay? And it's not until these stores are completely depleted that you start to impact on erythropoiesis. And of course, this is because the body wants to prioritize iron use for oxygen transport. So it tends to sort of preserve that at all costs. And so once we've depleted iron stores and you start to impact on hemoglobin, there can be a considerable lowering of hemoglobin before you reach clinical threshold that we define as anemia. And it's typically, for pregnant women, 10.5 to 11 grams per deciliter. Okay, so if you were to look at the left person on the left side of this graph would be considered somebody with normal iron status and no evidence of anemia. And then on the right side of this, where again beyond this clinical threshold, we consider them anemic. Okay, but there's an entire range in here in which we can have a depletion of iron stores without the manifestations of anemia. And we refer to this as iron deficiency non-anemic or latent iron deficiency. And this is important, of course, because you can still have a lot of the biochemical manifestations without the overt manifestations of anemia. And the reason why this is tricky is because, as I mentioned before, there's a lot of causes of anemia that could be unrelated to iron status. But detecting iron status is actually quite tricky. One of the reasons is because it requires a lot of tests. Numerous indices need to be used. They're usually difficult and fairly expensive tests. And the other thing that's worth mentioning is that a lot of iron biomarkers are actually acute phase proteins. And so that means that their influence are confounded by infection or inflammation. And so it turns out that diagnosing or identifying iron deficiency, much less without the presence of anemia, is actually quite challenging. And that's perhaps best illustrated right here. And this was just one study they pulled out where they looked at the prevalence of anemia among Inuit women in Canada. And in this population, 43% of the women were diagnosed with anemia. But then upon subsequent investigations, they found that really only half of them were attributed to iron deficiency. And even in those women that were recruited that didn't have anemia, again, there was almost an equal number that had iron deficiency, again, without concomitant anemia. So again, there's a lot of challenges, and the approach to diagnosis really has to be done carefully. Okay, so these are some of the challenges that exist in the population at large. And of course, when we're talking about pregnancy, and there's added challenge that we're dealing with 2 individuals and not 1. And health practitioners typically are reluctant to sample directly from the fetus, and because of the inherent risks it poses. And so rather what they do is they use biomarkers in the mother and they infer based on that evidence. Okay, but there is an implicit assumption there that whatever's happening in the mom is actually a good reflection of what's happening in the baby. And this really has never been tested empirically. And so we set out to do that a few years ago, where it was a very simple study. It was a clinical study where we recruited otherwise healthy women that were presenting for elective cesarean section at the Royal Alex Hospital. And essentially what we did at the time of their admission, we collected hemoglobin levels. And then usually a few hours later, at the time of delivery, we could collect cord blood hemoglobin levels. And then we just plotted them. Okay, and so again, I want to just remind you that these are otherwise healthy women that have no obvious pregnancy complications. And the first thing to notice is that in this particular 3% identified as or were diagnosed with anemia, according to the World Health Organization guidelines of 110 grams per liter. Okay, but the other thing that's worth mentioning is that there really was no correlation between maternal hemoglobin and cord blood hemoglobin. So this was all quite surprising to us. Now, admittedly, I just told you that, hemoglobin and markers of bioiron or biomarkers of iron status have to be looked at independently because they don't necessarily correlate and they're not necessarily a good reflection of one another. So the obvious question then is, how do maternal indices or biomarkers of iron status correlate with that of the fetus? So we followed this up with a systematic review, and this was done in collaboration with Maria, who's here. Nice to see you again. And a postdoc, Omalar Sami. And this was published in eClinical Medicine a few years ago. And so essentially we did a systematic review, and we collected data from a number of studies wherein they looked at maternal indices. So I've just shown a couple of here, but we did a fairly thorough examination of all hematological indices, as well as biomarkers of iron status, which are shown in blue here. And reported all the data that reported this in the mothers as well as in the cord blood, okay? And what I'm showing here in this graph is essentially the synthesized data, and it shows the R-squared values of the coefficients of determination, okay? And so I guess the first thing to notice is that there's a lot of gray boxes. And so in these studies, we just couldn't find any evidence of this correlation. So this work just hasn't been done yet. The red boxes show correlations that were done, but where there was no significant correlation. And so from that perspective, perhaps you wouldn't want to be relying on maternal ferritin to diagnose anything in the offspring. What's interesting is that even those boxes or those associations where we did see a significant correlation, by and large, they were very, very low. So the best we could do was maternal hepcidin, and the coefficient of determination was about 0.18. And that's not very good. Generally, even a poor correlation is considered, in the range of 0.5 to 0.6. Okay, so altogether, it really does suggest that maternal indices are really not a good surrogate for fetal indices, and that we really do need to come up with better ways of diagnosing iron deficiency anemia, because ultimately this is going to be what dictates intervention strategies throughout pregnancy, but as well as in the neonatal phase. Okay. Now, what's interesting is that when we were submitting the first clinical study, one of the reviewers came back to us and they were like, okay, well, that's fine. We recognize that there is no association between maternal and fetal indices, but how do you know you're more interested in the fetal outcome or the fetal indices? Could it be that perhaps, having a healthy mother, healthy hematological indices, healthy iron status is more important than for pregnancy outcomes, irrespective of what's happening in the fetus? And I think that was a valid point, and we don't have sort of a definitive answer on that. But what we did in that same clinical study that was conducted here in Edmonton is we had also collected placental weights. And so we did a brief correlation analysis between placental weights and maternal hemoglobin levels, and we also did it between cord blood hemoglobin. And you can see that there's this really nice correlation here, but not so in the maternal hemoglobin. And the way we've interpreted this is that, in general, the placenta tends to be a fairly plastic organ that's able to adapt and respond to the needs of a pregnancy. And in this particular case, it seems to be adapting to the needs of the fetus and the conceptus, rather than sort of the demands of the mother or the supply of the mother. And again, I think when you step away from it, it's probably fairly intuitive, but I think that this data really kind of really illustrates that nicely. Okay, so that's the diagnosis piece. So again, big issues with properly identifying iron deficiency and anemia, and being able to do so in a pregnancy where we can get a sort of clear image of what's happening in the offspring or in the fetus, without relying on maternal indices. So those are really the biggest challenges. The treatment issue I'm going to spend a little bit less time on, but there are a couple of points that I think are worthy of mentioning. And so the first one is that replenishment of iron stores really takes a long period of time. And so much like in the original iron depletion graph that I showed, where I mentioned that the depletion tends to occur over the course of months and years, I think iron supplementation and replenishment really has to sort of occur on a similar time scale. So it really has to be weeks, months. And the reason for this, or at least one of the reasons, is because in excess, iron can be toxic. Okay, and so if you do too much, ultimately the body will just not take it up. And excess iron, of course, can lead to accumulation in organs or tissues where you don't want it. And that, of course, can lead to dangerous free radical production or reactive oxygen species, which can actually cause harm. Okay, so this has to really be done in a gradual process. There's issues with compliance. So a lot of individuals that have iron deficiency anemia are given supplements, but whether or not the dosing is optimal, in general, it tends to cause a lot of GI discomfort. And so in pregnancy, when women oftentimes are not feeling great to begin with, these iron supplements just make them feel lousy, and oftentimes they just can't take them. Okay. And the third point, I don't think is unrelated to the first two, is that maternal supplementation doesn't correlate well with improved pregnancy outcomes. And so there was a couple of Cochrane systematic reviews that were published a few years ago, one of which was on daily oral iron supplementation in the mother, and another was on intermittent oral iron supplementation in the mother. And what they found was that while iron supplementation is great at alleviating the burden of maternal anemia, arguably more important outcomes like preterm birth, intrauterine growth restriction, and even perinatal death, the evidence was not as strong. So there was no conclusive evidence there. And so, I'm sure that this is an optimization issue. But notwithstanding, I think it really suggests that our current intervention strategies are perhaps not ideal for dealing with this for this global health issue that it is. Okay. So I realize I painted a bit of a dire picture here, and it really begs the question. Again, this kind of comes back to sort of deceptively complex. So despite the fact that it is obviously deleterious for, how come it's so prevalent, and why is it so intractable, and why can't we deal with it? And obviously we don't have a definitive answer on this point, but there may be some evolutionary insights that can sort of provide some explanations. Okay, and so this was a paper that was published I guess almost a couple of decades ago in the Journal of Nutrition. It's a really fascinating article. I really like it. But essentially, the argument's a concept paper, and the argument harkens back to what's known as the Neolithic Revolution, or the Agrarian Revolution, which happened some 10 to 12,000 years ago. And so this was a time that was characterized by the transition. Our forebears who were nomadic hunter-gatherer tribes, essentially to the transition of more agrarian-based societies. So this coincided with the advent of agricultural methods and the domestication of animals. Okay. And this happened over the course of thousands of years, and with it came sort of a number of important evolutionary milestones, not the least of which included, for example, dietary changes. So movement away from, for example, meats to more grain-based diets. And it was probably accompanied with a number of reproductive changes. So again, just the stability of these societies probably led to more pregnancies and shorter interpregnancy intervals. Okay, I'm focusing on these specific milestones because they probably contributed to the emergence of iron deficiency in pregnancy that we know today. Okay, but then sort of the obvious question still stands: why then didn't we co-evolve sort of other mechanisms that could improve iron uptake or reduce the health burdens associated with it? Well, it turns out actually that this transition was also associated with another important milestone, and I think that was the rise of infectious diseases. Okay, and so you can imagine then that in these societies with increasing population densities and cohabitation with animals, this ultimately led to the rise of zoonotic diseases. And this is important because there are a number of pathogens that are critically dependent on iron, not only for replication, but also for pathogenesis. Okay? And I've listed a couple of them here, and these ones are particularly important because not only are they dependent on iron, it turns out actually most pathogens are dependent on iron. These ones are particularly dependent on iron, but this is also a group that have exerted a particularly strong evolutionary pressure in our human history. So for example, Yersinia pestis is the pathogen that's believed to be responsible for the plague of Justinian and the Black Death that occurred in the 6th and 14th centuries. And during those time periods, the Black Death was associated with 1/3 to 2/3 mortality. It just killed vast numbers of the population. And so you can imagine then that any evolutionary benefit against these organisms could have had a lasting evolutionary benefit. Okay. And there's a number of other ones that are also implicated. And so this theory basically posits that under normal circumstances, if you look at the gray line here, that maybe mild, moderate, or even severe iron deficiency anemia would be associated with a reduction in overall evolutionary fitness. Conversely, if there was a strong selection pressure imposed by infectious diseases, then you might see an altogether leftward shift of this, such that mild and moderate iron deficiency might actually improve relative fitness. Okay, and again, it's worth mentioning here that it doesn't have to confer a huge evolutionary advantage, but as long as it's persistent over the course of a long period of time, that it could really have an effect in conserving that trait. And so we step away from this. I think it's an interesting hypothesis. And, the more and more I think about it, I don't really think that it's all that outlandish, because certainly even now we know that iron supplementation is not invariably beneficial. So for example, there are certainly some populations, for example, in sub-Saharan Africa where malaria is endemic. And in those circumstances, iron supplementation is not invariably beneficial for the mother as well, or the offspring. And so that's one of the reasons why it's a complex situation, but it's one of the reasons why the World Health Organization doesn't necessarily recommend indiscriminate supplementation across the board. Okay. So, and of course, even beyond sort of disease and treatments, I think it has important implications in other areas. So one of the areas I'm particularly interested in is understanding whether very preterm birth or preterm birth babies who are at most risk of iron deficiency and anemia are also at greatest risk of neonatal sepsis. So the question is should we be treating iron deficiency, or might it actually confer a benefit and promote beneficial health outcomes in some of these babies? And so we don't know that yet, but it's one area that we're interested in. But this data and this concept has really kind of like changed our thinking a little bit. And we've started to think about iron deficiency, though bad in many circumstances, that there might be some select instances where it may confer benefits. And so obviously I've talked about the inflammation, the infection. There's actually other areas as well. And maybe in the last 5 minutes or so, I'll just share with you a study that we recently completed. And that one really focuses on hypertensive disorders of pregnancy. So hypertensive disorders of pregnancy is a constellation of conditions, I guess, that include chronic hypertension, gestational hypertension, preeclampsia, and chronic hypertension with superimposed preeclampsia. Okay, and so in general, collectively, they affect about 6 to 10% of pregnancies worldwide, and they are a leading cause of maternal mortality globally, but in particular also in industrialized countries. Okay, and perhaps what's most disconcerting is that the prevalence is actually increasing. And so coming back to iron deficiency, I haven't really talked about many of the biochemical consequences, the functional consequences, but when we're looking at cardiovascular function, it turns out that iron deficiency does some good things. And so a number of studies have published that iron deficiency and anemia are associated with reduced blood pressure, increased cardiac output. And the thinking here is that with anemia, and again, with reduced oxygen carrying capacity in the blood, the heart tries to adapt to grow a little bit so it can compensate, it can pump out a little bit more and try to overcome that deficiency that's called pump a little bit better. Importantly, it turns out that reduced anemia is also associated with increased vasoprotective nitric oxide bioavailability. And in fact, this increased nitric oxide signaling within the vessels is believed to be one of the major mechanisms by which anemia reduces blood pressure. Okay? So looking at some of these functional outcomes, the obvious question is might there be some circumstances where iron deficiency might confer cardiovascular benefits? And the obvious one that we've been talking about is in hypertensive disorders of pregnancy. So this is what the question that one of my graduate students, two of my graduate students, Jad, Julian Rashid, and Jenny Vu, opted to interrogate. So to do this, we used a rat model, and to mimic a hypertensive disorder of pregnancy, we used the spontaneously hypertensive rat. So this is a rat model that's been around for no less than 50 years or something. And ultimately, through a number of breeding experiments, they developed a number of random mutations that led to the spontaneous emergence of a fairly profound hypertension in early adulthood. Okay, so whereas a normal rat would be the same as a human, 120/80, SHRs develop hypertension probably around 180 to 100, and 160 to 180, I guess, at around 12 weeks of age. And so we've got the original strain from which the SHRs derived from. And so these are our normotensive control. And so in this particular study, what we did is we took our hypertensive animals and our normotensive animals, and one group we gave a control diet, and the other group of both, we fed them an iron-restricted diet for 4 weeks before pregnancy. So this is gestation being -28. And then we maintained them on that diet throughout pregnancy, which is 3 weeks in this rat. And so I'll just show you a little bit of the data here. So this is what happens. I haven't labeled these, but it doesn't really matter. This is the rust color are the iron deficient group. And you can see that now with this progression, or with the progression of pregnancy, you see this progressively worsening anemia, okay, that culminates to what we'd probably consider a moderate anemia by the end of pregnancy. As I mentioned before, you can't just rely on hemoglobin. We have to know the underlying cause. So we looked at a number of markers of iron status. And so you can see in this particular case, this is the transferrin receptor mRNA. This goes through the roof. This is a major transport protein that upregulates. And so when iron is deprived, you want to increase transport as much as you possibly can to restore those iron status. So you can see this goes through the roof. So we're seeing a clear evidence of iron deficiency anemia. Okay, so that's in the mom. What happens in the fetuses? Now, I'm not going to spend a lot of time in the fetuses, but I do think it's worth kind of just showing you that what we're seeing in this animal model does recapitulate what we see in humans. But despite this sort of modest iron deficiency, we see a fairly severe anemia in the offspring. And again, I think this really just highlights that point. There is this discrepancy, this disparity between hematological indices or severity of iron deficiency anemia in the offspring compared to the moms. So I wouldn't rely on this to make a judgment about this. It causes body weight restrictions or birth weight restrictions, so intrauterine growth restriction. And I know, it's tempting to speculate that it's a greater reduction in the normotensive animals compared to the SHRs, but I'm not 100% sure. But it does cause growth restriction in general. And you start to see in the offspring a lot of the physiological adaptations you might expect. So, for example, this increased heart rate. And again, this is again an adaptation where the fetus even wants to overcome the reduced oxygen carrying capacity. So the heart gets a little bit bigger, pumps a little bit stronger to overcome that oxygen deficiency. Okay. So that's the model, and it seems to work quite well. And so the real question for us was, what about cardiovascular function in the dams? Okay. So we looked at systolic blood pressure both through pregnancy and then afterwards, and we confirmed this using 3 different ways of measuring blood pressure. So this is the tail cuff data that shows the nice profile. But you can see that in the iron replete SHRs, they're quite a bit higher than the control with Kyoto, again, as you might expect with a spontaneously hypertensive animal. But if you look at the iron deficient animal, the iron deficient SHRs, you can see that with the progression of anemia, you see this gradual lowering of blood pressure that ultimately reaches its maximum at the end of pregnancy. So we thought this was really interesting. And in fact, you see the similar effect, albeit not the same magnitude in the Wistar Kyotos. So again, with iron deficiency and the progression of anemia, you start to see this blood pressure lowering effect, and it's shown perhaps best right here. It's a little bit of a lowering in the Wistar Kyotos, but it's much more pronounced in the SHRs. Okay. Now, at first glance, that seems great, but the other thing to consider is that blood pressure exists for a reason, and that's to push blood through organs and through various tissues. And so if you reduce the blood pressure, the obvious question is that impairing your ability to perfuse organs with blood? And so to test that, we also looked at uterine iron blood flow. Okay, and we were delighted to see that there was in fact no changes. And even though we have a reduction in perfusion pressure here, one of the reasons why it doesn't affect blood flow, we think, is because, as I mentioned before, one of the ways it's doing so is by actually causing a vasodilation of the blood vessels. So you're reducing the vascular resistance within those blood, with those regions. And so it just means that you don't need the same pressures to actually effectively push blood through those sections, those tissues and regions. Okay. So this all looks good from a hemodynamics point of view, but as I mentioned, one of the big compensations is the maternal heart. And so that's what we did next. And obviously, I'm just showing some highlights of this study. This is my, I guess, last slide here. But we were interested in the heart because, again, if the heart has to work harder during pregnancy, it might actually cause underlying deficits or dysfunction. And so we looked at heart weights, first of all. This is the body weight. And you can see that the iron deficient animals have increased heart weights, much like the fetuses. But this is mostly in the Wistar Kyoto, the normotensive, and we don't actually see this come out in the SHRs. I'm not 100% sure why that is, but you'll see that to begin with, the SHRs actually have bigger hearts to begin with. And so it might very well be that the bigger heart, the muscle is already there, and then when suddenly you lose or you drop the blood pressure, they don't require the same kind of compensation to actually increase the cardiac output that you see. Okay, but we looked at diastolic volume, which is essentially the chamber size of the left ventricle, and you can see that there is cardiac remodeling. So the chamber sizes get bigger, even though the hearts don't. Okay, and so altogether, we think that this might be a way of improving cardiac efficiency. And so this was done by echocardiography. And we also did pressure volume loops. And one of the parameters that you can assess is looking at the potential and the used energy and calculate these ratios. And you can find that you can calculate cardiac efficiency. And whereas none of the other groups, whereas the SHRs, the iron replete animals, seem to be impaired cardiac efficiency. Perhaps not surprising, given it is a disease model. The efficiency really goes up with the introduction of the iron deficiency and anemia. Okay, so it suggests at this stage, at least, that the heart is working a little bit better. And to confirm that, we looked at some circulating biomarkers that are typically used as a marker of cardiac stress or cardiac injury. And we found that there was no real changes with the sole exception of the iron deficient hypertensive animals. And so it seems overall the injury is reduced in these hearts. So that's all I'm going to show about this. But I guess want to make the note here that that by no means, based on this data, should anybody stop taking their iron supplements or stop eating, or stop seeing their physician when there's iron deficiency, because there's a lot of caveats to this. Certainly one of them is that this is done in rats. The other thing is we've only looked at functional outcomes, and so we're also assessing the biochemical effects, but we don't really know what it does sort of on a tissue basis or an energy metabolism level. And the other question is whether or not these benefits persist after prolonged exposure. It might very well be that the benefits are seen for 3 weeks of pregnancy, which characterizes a rat pregnancy, whereas maybe after 6 months. You get decompensation of the hearts. And so then the heart can't cope with it after a prolonged period of time. And so this would absolutely have to be tested in humans as well before we can sort of make any definitive conclusions. Okay. But there is one more point to be made here, and I'll just kind of bring that up afterwards. And so just to summarize everything that I've said so far, I think iron deficiency is a highly prevalent, but it's a deceptively complex global health issue. And I would suggest that the current methods for assessing fetal and neonatal iron status and anemia are inadequate, as are current intervention strategies. And so a lot of work, I think a lot more work to optimize treatments and diagnoses really need to be done here to improve if we're going to make sort of any progress in reducing the burden of anemia globally and meet the sustainable development goals. Okay. And the last point I mean is probably a little bit controversial, but iron deficiency may actually confer some health benefits in some pregnancies, or at the very least mitigate the severity of other health complications. And from that last study, this might be the most important point. It might be that while we don't necessarily recommend iron deficiency anemia as a sort of a therapeutic or a treatment option, it might very well be that it keeps other pathological outcomes at bay. And it might be that, for example, a woman who is iron deficient, if we start to give them iron supplements, it may actually reverse the iron deficiency, but it may in turn also unmask a hypertensive phenotype or preeclampsia, for example. And so, in fact, there is a lot of studies that have shown that one of the side effects of iron supplementation is actually hypertensive response. So I do think that this is worth thinking about, and I think it adds one more layer to this complexity of iron deficiency as a global health issue. And so, arguably, this is my most important slide. So I especially want to thank my team, and in particular, shout out to Jad and Jenny, who were largely responsible for the data that we collected in the SHR study. But I also want to thank the collaborators who were involved in the study, and also I have to acknowledge the funding agencies. And so with that, I'm delighted to take any questions, and thank you very much for your attention.
Thank you very much, Dr. Bourque. That was a very interesting presentation. I have some lived experience with iron deficiency anemia in pregnancy, and treatment of it, and not a lot of success. So it's really interesting to hear the whole health kind of perspective on it. Same process as with Dr. Kozyrskyj. If anyone has any questions, feel free to raise your hand, or you could just chime in or put your question in the chat, and I'll do my best to read it out. Rhonda, I see your hand up. Thank you.
Sorry about that. There we go. Here we go. Okay. Anyways, you can hear me, I hope.
We can, yes.
Okay, good. Thanks. And thanks for a great talk, Stephane. And yeah, for going into so many different questions, but I'm going to try and confine my questions at the moment to just things. One is I was just wondering, there's quite a lot of excitement about thinking about iron, its different forms of iron, heme and non-heme, as two separate nutrients, actually, because their intake, absorption sources, and even transport and distribution may be quite different. And as far as I know, most of the work that's been done in iron supplementation has used the non-heme form, which is tightly controlled, but the heme form has been less well studied. And my first question is around what you think about optimizing iron status prior to pregnancy using heme forms rather than relying on non-heme forms, which we know don't work well during pregnancy. And we have hardly any interventions that, once initiated during pregnancy, actually work during pregnancy. So I just wondered about your thought on that.
Yeah, and I presume you mean sort of from a kind of a therapeutic or supplementation point of view, but not necessarily relying on increased red meat consumption, animal products. And I think the biggest one there is cost. I think, one of the benefits we think about for iron supplementation, just regular supplements, is that they're sort of really cheap, and so that's typically the first. And my understanding is that a lot of the heme-based iron, they're quite expensive. Yeah, and so but I think that, like, the bioavailability, you're absolutely right, and the transfer is much more bioavailable. So I think it's 4 or 5 times, whereas I think, like, inorganic iron, I think it's maybe 10% bioavailable, whereas the heme is probably 2, 3, 4 times that. So I think that is definitely an option. But again, yeah, I'm not sure what in terms of market availability and whether it is there. I guess the other ones to consider are iron, intravenous iron. They've got a lot of new complexes that seem to be doing quite well, and I think they've overcome a lot of the challenges associated with, for example, like the anaphylactic reactions, and the other one is blood transfusions. But I think just by virtue of the fact of the healthcare commitment that's required to actually institute those treatments, it all comes down to cost again, and it's just not a cost-effective way of dealing with the population on that scale.
Yeah.
Does that make sense?
Yeah, and I guess, I guess there is that other element that we sort of took off the table right off at the beginning, but that was really around diet, like optimizing diet prior to pregnancy and what that actually means. Because it seems like there's a lot of things, like you could take folic acid, for example, where we know a lot about folic acid supplementation, but it really does need to be prior to pregnancy to
Yeah.
get full benefits, right? And I'm
Yeah.
wondering whether there might be some work that needs to be done looking at optimizing iron status, in kind of in a similar way. And I see that the message in the chat from Lisa. Yeah, that's a really great point too, Lisa. So yeah. Anyways, thank you very much. And Stephane, I know we'll be in touch soon.
Yeah. Let's have coffee.
Thanks.
Okay, I see Lisa has a question in the chat. Until this year, the WHO recommended using capillary hemoglobin testing with the HemoQ. This has recently changed, and now a venous sample is recommended. In Africa, should we stop using our HemoQ devices? We do a lot of field work and refer for further evaluation based on capillary hemoglobin and would not be able to do so with venous analysis. What would you recommend?
Yeah, I mean, I don't work mostly with clinical populations, and so I certainly wouldn't make any recommendation. I think there's a lot of other people who could comment on that, I think. But from our own experiences, even in the lab, we've noticed discrepancies as well between using capillary blood as well as venous blood, and there is a mismatch. And in fact, we do use the HemoQ because it is convenient. You can carry it almost anywhere, that you can plug it in. And so it is, I think, a very useful tool, but we do have to interpret that data with caveats, recognizing that, it's perhaps not the best sample. So, recognizing that, there might not be other options. I think some screening, even if it's not the best screening, is probably better than no. But again, as I mentioned before, I think that the anemia piece is really only half of it, and there has to be better ways of identifying the underlying causes, because that's really what's going to dictate the management and intervention strategies. And so I don't know if there are easy sort of point-of-care systems where you can assess iron status, but yeah, I think that's a really important component of it as well. So I don't know, I hope that answers your question, Lisa.
Okay, Lisa says thanks. Dr. Kozyrskyj, I see your hand up.
Yes, Steph, I really enjoyed your presentation, especially the material on the the evolutionary benefits of iron deficiency, and I'm really pleased with the kind of a newer direction that your research has taken to kind of start to test some of our, I don't know, I guess common practices to when something is low, you measure something in blood and it's low, and then you try to fix it, with the supplement. But you and I have tried to collaborate on iron, right? Iron supplementation during pregnancy from this perspective of harmful effects on gut microbiota.
Yeah. That one is sort of a whole other level of complexity, because I think it really comes back to, I think certainly iron status in general could be an important contributor to to the microbiome in general. Whether or not it contributes to dysbiosis, I think that I recall seeing a few of those studies, and I remember seeing some of the pilot data that we looked at. I looked at that fairly recently, and I think there were some sort of interesting sort of patterns that emerged in our principal component analysis. So iron deficiency certainly seems to be doing something. But as you and I have talked about, I think the other piece is really relates to the suboptimal treatment, and so if the knee-jerk reaction is to just give as much iron as we possibly can, recognizing that the body is not equipped to properly deal with it. A lot of it just a lot of the iron supplement ends up in the GI system. And of course, that ends up being sort of an ideal platform for some of these more pathogenic bacteria. I think you had mentioned it's the enterobacteria that seem to be sort of more dependent on the E. coli, E. coli that seem to be dependent on the iron. So we could be producing sort of an environment that's conducive to bad bacteria growing. So that might be one of the reasons, even independent of iron status altogether. And yeah, I think it's another kind of aspect that I really very much like to. So we should start up those conversations again.
We should restart the conversation. Yes, we did apply to WICRI to pursue this research question. Hi, for anyone who's listening.
I hear you, Anita.
Right.
Try again.
How many times should we try? Right.
Okay, so I see we're at 1 o'clock. I just want to thank you, Dr. Kozyrskyj and Dr. Bourque, for your presentations today and for everyone joining us today. Everyone who is participating today are encouraged to contact wcgrants@ualberta.ca or stay in the Zoom space to talk to Michelle if you're interested in participating in this research cluster. Yeah, it was a great session. Thank you so much.
Thank you. Thanks for your questions.
Thanks, everyone.
Have a good week.
Thank you.
Bye.